Hello, everyone. We're going to take just a minute and let everybody get on for the webinar today, and then we'll go ahead and get started. Thanks for joining us. All right, I think we'll go ahead and get started. We'll let a few more people trickle in as they come. Hello, everyone. I'm Kirsten Wakefield, the Stakeholder Engagement Specialist for Maricus. Together with Dr. Janet Reamer, Science Program Director for MARCO, we co-coordinate the Mid-Atlantic Coastal Acidification Network, MACAN. Thanks so much for joining us for the final webinar in our spring series. Today, we'll hear about new ocean acidification research spanning the Northeast Atlantic to the Chesapeake Bay. The first two talks will highlight different approaches to studying socioeconomic vulnerability for the sea scallop fishery and for Chesapeake Bay oyster growers. Our next two talks will highlight the role of modeling and genetics in analyzing the impacts of OA and other climate stressors on oyster growth and habitat suitability. Before we introduce today's speakers, I'd like to make a quick plug to save the date for MACAN's biannual State of the Science workshop. It will be held in person September 11th through 12th at the Institute of Marine and Environmental Technology in Baltimore, and we really hope to see many of you there. It's the first in-person event we'll be having, over the past couple of years, so we're pretty excited to be hosting again in person. And now today, I'd like to give a warm welcome to our four featured speakers. The first is Dr. Samantha Siedlecki. She's an assistant professor at University of Connecticut. As an oceanographer, she employs numerical simulations to identify processes in coastal regions that drive the biogeochemical dynamics in modern and future oceans. She has recently begun to explore regional climate projections of ocean conditions on the east and west coasts, and she partners with social scientists to bring these tools into decision-making frameworks. Today, Sam will share her research on a dynamic pathway to transition from vulnerable to resilient fisheries social ecological systems, a transdisciplinary case study of the U.S. Atlantic sea scallop fishery. Our second speaker today is Brian Katz. Brian is a human geographer and geospatial scientist from Oregon State University who applies geovisualization and community engagement methods to inform climate adaptation planning efforts aimed at reducing vulnerability and building adaptive capacity equitably. Today, Brian will share their interdisciplinary research on sustaining Chesapeake Bay oyster livelihoods in the face of OA and multi-stressors. Continuing with our focus on the Chesapeake Bay, our third speaker today is Czajka. She's a master's student at the Virginia Institute of Marine Science, working with Dr. Margie Friedrichs and Dr. Emily Rivest to examine climate impacts on local carbonate chemistry and oyster growth. Today, she'll be discussing how she's using a 3D coupled hydrodynamic biogeochemical model to examine impacts of future warming and atmospheric CO2 projections on oyster growth, and how that information can be used to identify suitable areas for oyster aquaculture and restoration. And we'll finish up today with our fourth speaker, Annie Schatz. Annie is a PhD candidate at the Virginia Institute of Marine Science. She works with Dr. Emily Rivest to study the potential physiological impacts of climate change on the early life stages of marine invertebrates. Her research focuses on effects from OA and multi-stressors and how environmental history may impact performance of later life stages in shellfish aquaculture. Today, Annie will share her findings on larval resilience to climate change in wild versus selectively bred aquaculture broodstock of the eastern oyster, Crassostrea virginica. The format for today will be 4 15-minute talks followed by 20 minutes of Q&A, and all participants will be muted, so please ask your questions via the Q&A box at the bottom of the screen. Anthony Himes, our Sea Grant OA Fellow, also from the Virginia Institute of Marine Science, will moderate the Q&A discussion for us today. And just a reminder that this webinar is being recorded and will be available on the MACAN website on the resources page. So without further ado, let's go ahead and turn it over to Sam.
Great. Can you hear me and see that okay?
Yeah, that's perfect, Sam. Thanks.
Perfect. Great. Okay, well, thanks for having me here today. I am excited to talk to you about our regional vulnerability assessment project funded by the Ocean Acidification Program. And the three of us, myself, Shannon Masek, and Lisa Colburn, are the co-leads of this project. And there's a large team of contributors, the PIs of which and their affiliations are listed on this slide. Shannon Masek, is a specialist in bivalve physiology and works at the Noah Milford Lab. And Lisa Colburn has a broad range of experience in social science and has been lead developer of several of the community vulnerability indices used by NOAA fisheries. And so together we've been leading this effort to kind of to develop a regional vulnerability assessment, specifically this dynamic pathway to focus on the sea scallop fishery on the east coast of the US. And I'm Samantha Siedlecki, and I'm a modeler. So my expertise is in biogeochemical models. So on this East Coast, I just want to start off with, some of this new result that was now highlighted. Ocean acidification is highlighted, and the state of the ecosystem report, the New England report that came out. And so here you can see that the subsurface conditions already are already experiencing seasonally low saturation state values in the Mid-Atlantic Bight and the Gulf of Maine regions. I'm not defining that here because we are in a CAN space. And so I feel like this ocean acidification term is well understood, but please, if there are questions about that, let me know. So here on this one plot here showing you summer conditions from multiple years, showing you that observationally we're seeing over the summer regions where repeatedly saturation states are low, and so the colors here indicate lower values than 1 in the lighter colors and greater than 2. These are just bottom conditions, just summer multiple years, and the dots indicate sort of in situ hydrographic observations, bottle samples. But the more continuous paths you see there in both the Mid-Atlantic Bight region and the Gulf of Maine come from these gliders that the Grace Saba's group has been putting out. And she's the one who generated this figure. One of the panels I'd just like to draw your attention to is this hot spot region that was identified in the Mid-Atlantic Bight region for sea scallops here in the report. And so this depth range here is between 25 and 200 meters. Those green dots are highlighting areas from a subset from this map, showing you the aragonite saturation states that are lower than 1.1, which they identify in this report as a sensitive range for the Atlantic sea scallop. So we know that these conditions are already happening, in our current state. And why is that? Why does that matter? Well, over the past many decades, the fisheries in New England and Mid-Atlantic have become increasingly dependent on scallops. And so you can see that here. This is a figure from my 2021 paper that was updated from Dwight's paper from 2015, showing you those purple bars of getting larger in size as you move forward in time. This values here are like 1958 through 1968, spanning all the way through 2018 over here. And so the communities in this area, this mapped area here from a report from John Hare from this region, are increasingly dependent on sea scallops. Jenny Rubin had a really nice paper from 2018 where this figure is from, talking about how, what this figure portrays is kind of some background about sea scallops for those of you who don't know, that some things I was surprised and sort of learning about in writing the proposal and thinking about this project is that scallops live for a very long time. So for example, they can live for up to 20 years or so. And so that means that if we're thinking about projections out to 2100, that really is only traversing about 4 generations from now, right? So it's really not that long. Adults grow, here, this cartoon showing you on the bottom, of course, for about 4 or 5 years until they reach harvest size currently. So it takes them a good while in order to get to the size where they're harvestable. And of course, what's also depicted here is that the larvae and the adults occupy different regions of the water column. So the larvae are floating around in the surface waters, experiencing those conditions, which may be different than those at the bottom. And then, of course, the fishery is also an important forcing on this, and to be something to be considered over this time period as well. So the fishery, what's happening with it? This is a plot showing you harvest in millions of pounds from 1990 through present. This is data available from NOAA Fisheries. And so what you're seeing is that the orange is showing you the New England harvest, and then the blue is showing you the Mid-Atlantic harvest. And what you can see is that the harvest is changing over this time period, and that seems to be a shift in it to the northern regions and more into the Gulf of Maine area, into the New England area over this time range. The other forcing that I talked about, the fishery, is also important to sort of take into account, and the management of that fishery over this time period. And so the formal managed rotational closure program that we understand and know today began in 2004. And so the New England Fisheries Marine Science Center closes areas with large concentrations of small, fast-growing sea scallops to protect them. That's the motivation, is finding the small beds that are of small, large, fast-growing sea scallops in order to protect them. And that's been shown, in some work that Devora led, showed that to boost sea scallop meat yield and yield per recruit. You can also see that 2004, time period kind of, as is notable on this time series. So, what's causing this trend? There's, of course, notably some ocean warming going on, in the area, which we've all also heard about, hopefully. There's a lot of warming going on out there, but there's also these ocean acidification patterns and trends, and of course, changes in sort of the management structure of the fishery and sort of what's driving these changes. So the main project objectives that we're working on are to sort of determine the vulnerability of the sea scallop fishery to ocean acidification and temperature changes. And then the second one, sorry about that, there should be two, not one. Looks like a little conversion happened when I converted this from Google Slides to PowerPoint. But investigating ways, the second goal is to investigate ways in which the fishery could choose to build resilience to these projected changes. And so we're also working with the fishery through workshops, which I'll talk more about. So how are we going about this regional vulnerability assessment? Well, first, we're working with a lot of the natural science information to determine the scallop risk and think about some of the physiology questions, to project the ocean conditions locally into the future, to combine that risk information with the ocean condition projections, and then actually predict shifts in biomass as well as time to harvest within this region. At the same time, social science team is working to assess trends in historical fishing, to quantify risks to coastal communities and determine their adaptation options, as well as examine the community response to closures in particular. And then as a group, we are working together to see if there's relationships between the spatially in particular, between these ocean trends and the fishing trends. We have an advisory committee that we report to and and discuss our results with. We also do a series of workshops with the fishery themselves, and where we communicate risk and identify future strategies. And in the end, we're hoping to generate recommendations to promote future resiliency. But the reality is that this is not just a once around kind of process. It turns out that we are learning from the workshops and going back to the natural science and bringing that. And so it's kind of it's a loop, really, more than anything. So we did workshops in two series now, both in 2021 and in 2023. Those included New Bedford, Point Judith, Barnegat Light, Newport News locations. And we found that there were a lot of questions about scallop physiology and ocean acidification impacts. We found that the fishery was open to changes in management actions. For example, like longer or change closures in that rotational management structure in order to sustain the fishery. We found there was a general skepticism for model projections out that far into the future, which is not uncommon. And there were discussions about the changes observed by the fishermen to the shells, as well as where they are fishing. And then there was a series of follow-up conversations with workshop participants and as well as others. And then in the end of the first round, a flyer was generated. And in the second round of workshops, we're working on generating a summary report that, because we just finished the workshops, the second round a little bit ago, and that will be distributed to the workshop participants. The flyer that you see just popped up here on the left was a field guide to observing impacts of OA. And this was meant to, basically, if there was some shell discoloration, some color changes, some things that other bivalves also experience, but that Shannon's group noticed in the lab conditions. If they found those kinds of conditions, we wanted to know and give them a pathway to engage with us about that. And so now we have one more round of workshops scheduled in 2024, and so we look forward to those. So one of the things that we have been working on in part of that loop, that feedback, when we learned the first round that there was some reticence about the models that far out, is that we actually tried to rely on observations collected by the fleet in order to evaluate the simulations. So here, there's a project that the Commercial Fisheries Research Foundation has in collaboration with WHOI. That's like a shell research fleet observational data set. And so this little map on the bottom here shows you locations where fishermen took CTD profiles as part of this project. And you can find more about that on this website. And then we have this historical simulation that spans from 1980 to 2015. And so we took the observations that was served as a base for the future projections. And so we took these fisherman-derived observations and compared those to the regional simulations, and then showed these observations in the workshop in an effort to build trust with the community. And then we show them, we go forward in time now, obviously, so the CTDs are only equipped with temperature and salinity, and so those are all we have to compare with. And then, as we move forward and show them projections, we move into these ocean acidification variables. But one thing that is, so it would be great to have other variables, that could be collected on those CTDs if that was possible. But one thing that's striking from the model projections is that there's this latitudinal pattern in the future ocean conditions. So for example, on the left here, what's plotted, now this is a continuous simulation from 1980 through 2100 under forced with the most extreme SSP5 8.5 from CMIP 6, so the most extreme emission scenario. And what you see here is that the year when the saturation state for aragonite is below 1.5 for more than 6 months of the year, so the majority of the year. And the year is the same color bar for each of these maps. And so the blue colors indicate closer to now, and then the yellow colors indicate far out into the future, more like 2100. And then for because we're trying to communicate with the fishery, we put some markings on there that would help them to orient themselves in terms of space. And so those include the rotational areas, both past and present. Some are closed, some are open, as well as the EEZ, because between the US and Canadian waters, they're in red. And so what you can notice is that the Gulf of Maine and the regions further north in these bottom conditions are experiencing those conditions now, right? You can also see that area that Grace's group has identified in that with the repeated observations off of Hudson River there is also experiences this earlier in the century. kind of more than 6 months a year, or majority of the year exposure. And then in terms of on the right, what you're looking at is temperature, which is of course the other important factor to be considered here. And that temperature is above 18 degrees C just for a month, right? And so if you're just over that for a shorter period of time, because this is a lethal threshold, Shannon tells us, for the adults. And so we wanted to be, we tried to give that a shorter exposure time. The exposure time for saturation state, we don't know as well. So here you can see that those conditions are mostly in the south and in shallow areas on Georges Bank, and they're much earlier in the century. So this kind of gives you the idea that there may be some benthic habitat compression, that the saturation state signal is kind of moving from the north to the south, and then the warming is coming up from the south. So we combine that with some results coming out of Shannon Masek's lab that were published earlier this year, led by Emilian Bus et al. In 2023, which showed this kind of interactive nonlinear effect of the combination of PCO2 and temperature, such that on growth. And so that's denoted by cartoon scallops here. So the growth rate is proportional to the size. And so what you're seeing is that, as you get higher in CO2, despite the fact that, so at modern conditions, they grow faster as things get warmer. But then as you get higher in CO2, there's like a level where that stops happening, and that CO2 effect starts to take over. So Halliburger, who's a student working at UConn with the team, has been developing a dynamic energy budget model, and taking that in order to bring those physiological measurements from the lab into the spatial management context. So here's an example of her preliminary work showing from just Georges Bank, forced by that dynamic, by the ocean model that I just showed you the results of. And so here she's just comparing, some results from the beginning of the simulation and the end of the simulation to kind of highlight the change. And what you see is that that the model is projecting that there will be a smaller ultimate growth length in the future. So they might be smaller as a result of that, which of course would matter for this decision of harvest size. So kind of time to harvest. And we look forward to Hallie's continued results and that spatial variability of how that will change in the future. So the social science team has been taking their tools and applying them to this issue, in particular, understanding adaptation to past events. And so they're reviewing the regulatory history of the fishery, analyzing fishing effort over time at the vessel and port level, and conducting oral histories with the stakeholders, focusing on history in the industry, adaptation strategies, the current conditions and future projections, and then developing scallop community profiles. So here's an animation showing you an example of some of the data they're working with as well, which includes a change in fishing effort over time. And so what you're looking at is a movie of where the communities have been fishing over time. And the next step now is to take these data sets and combine them with the ocean trends, and sort of see and then focus in on some of these events that are where there's been some closures, and examine the community response, both through those interviews and oral histories, as well as with these data sets, and specifically in response to some of these closures. So some examples of recommendations that could emerge include suggestions for future rotational closures, strategies and locations and durations, and the knowledge about which life stages are vital to protect and from which stressors in order to inform action. So I'll leave you with, it looks like I'm almost about out of time. I saw Kirsten come on, but my conclusions are basically that despite the masking of the OA signal in the region, by continued warming. The projections indicate the majority of the year will experience stressful conditions within benthic habitats in the Gulf of Maine by 2050, but it'll be later in the mid-Atlantic bight. That warming and OA stress may cause some benthic habitat compression in the region. That preliminary results from the dynamic energy budget model indicate that future ocean acidification and warming will cause scallops to grow, will ultimately reach a smaller size with potential reproductive implications. And there's spatial variability in fishing effort over time, which will provide us with a footprint of the fishery itself, and corresponding to these changes in the ocean conditions. And it will also make our results locally relevant and connect with the communities that where we're doing the workshops and interviews. Workshops with the very engaged fishing community have helped identify some key areas of concern and provided some educational opportunities and resulted in this flyer as well as this project summary report that we're able to produce and spread the word. And there's more workshops planned 2024, so stay tuned for those. With that, I'll just thank the funding agencies and happy to take questions. If you want to find out more about the project, you can go to the QR code website here.
Great. Thank you very much, Sam. If anybody has any questions for Sam, please type them in the Q&A box now, as she has to hop off early. But I had a question kind of to get us started. From your the workshops you had with fishermen, did you kind of get a feeling of, were the fishermen already really well informed about OA and concerned about it, or was there still kind of an education component there as well?
There was definitely an education component. They were more concerned about warming and about the wind farms, and we continue to hear about the wind farms also as a concern, from them so there are lots of things that they're concerned with, right? And so really the question is where to put OA, and the challenge that we've faced is the lack of an immediate threat in a lot of places, that it's more of a planning out, longer term. And so, we've had to kind of pilot out some of our communication strategies around that in order to help inform, why we think it could be an issue and when,
Okay. I guess we're a little tight for time. No questions have come through yet, but if any do, we can pass them on to you. So with that, I'll turn it over to Brian.
Great, thanks so much.
Great, thanks. So hi everybody, I'm Brian Katz. Let me go and share my screen here. It's loading. Okay, can you all see that now, hopefully?
Yep.
Great, thank you. So I'm Brian Katz. I'm from Oregon State University here in Corvallis, Oregon, but I'm on a team of many amazing scientists, some of which are on the call today. You'll hear from Catherine right after me. We call our team the Shellfish Thresholds and Aquaculture Resilience Team, or the STAR acronym. But it's a team of modelers and biologists and social scientists and some partners with the industry and restoration side of the issue. But today I'm going to be talking about oyster livelihoods in Chesapeake Bay. So just a brief outline, a big background, go into my framework and methods, results, conclusion. So when it comes to Chesapeake oyster livelihoods, I borrowed this analogy of the three-legged stool from Andrew Button at the Virginia Marine Resource Commission. He describes Chesapeake Bay as having these three really key sectors that kind of all hold up the system of oysters in Chesapeake Bay. You have the commercial aquaculture industry, you have the restoration partners, and you have the watermen, who are folks who harvest public oyster reefs for their livelihood. So these three sets of actors all comprise the greater system that we're interested in studying. When it comes to the multiple stressors, we've heard a little bit about OA already. We know that's a stressor that oyster livelihoods have to cope with. Also, extremes in salinity, both high and low salinity, can be a stressor that can impact the system. High and low temperature, high and low oxygen. These are all stressors that can combine to produce what we call social ecological system thresholds. And you might think of a common threshold that's often cited as like stressors combine and lead to oyster die-offs. But there could also be some social thresholds, like the decision for a long-time waterman or oyster grower to exit their oyster livelihood after a certain threshold is reached. So these are the types of questions that our project is trying to answer. So where and when and how do these thresholds manifest? So this is our framework that I'm using. I won't go into it too much for this presentation for lack of time, but I will just highlight that this is a social ecological system model where the rectangles here on the lower left are the physical elements of the system. The ovals are more of the human elements of the system. And you have all these arrows that connect every aspect of the system. So the main research questions that I'm answering with this research pertain to this section down in the lower left with regard to these exogenous drivers and shocks that affect the resource. One question I'm asking is where and when do changes occur in the social ecological system, or SES? How are these changes explained? And then more broadly applied to the whole system, how does the system respond or adapt to changes? And who are the key actors and assets needed to respond to changes? The methods that I've used for this research first includes geovisualization, meaning I'm mapping the best available biophysical and social data that we have from the top down. Then I conducted some spatialized interviews, originally on Zoom during the pandemic, and then more recently last summer in-person fieldwork in Virginia and Maryland. And those spatialized interviews were aimed at detecting and attributing changes from the bottom up. And then lastly, I performed a random forest regression, which evaluates the effect of multiple stressors on aquaculture production and assesses the relative importance of the different stressor predictors. And so just to give you kind of a quick glimpse of each phase that I just presented here, with the geovisualization tool, it's a web map. This is just showing you layer by layer, so bear with me here. The first set of layers pertain to the social system. You have aquaculture leases and restoration sites that are shown on the map here. But then when it comes to the biophysical data, we've got this really amazing biogeochemical model that Catherine will speak more about in the next presentation. But it models bottom conditions for temperature, salinity, oxygen, saturation state, total suspended solids, and particulate organic carbon, POC. But then we also have the biologists on the team, Emily Ribas and Mark Brush, who have compiled species sensitivity curves to those multiple environmental parameters. And so when we put them all together, we come up with maps like this. This one is showing the scope of growth for an average size oyster, averaged over 1985 to 2020. So it's an INCAS model. But these are just the kind of red areas are where oyster growth is the lowest on average. And the yellow here is where oyster growth is the highest on average. So then in addition to the growth function, our team also came up with net calcification functions, which describe whether shells are more likely to form or dissolve based on the temperature and saturation state conditions. So similarly, we mapped that function onto this map to show where red is where the conditions are more likely to dissolve shells. See that upriver for the most part. And then the blue is where shells are more likely to form, which we see in the open ocean and a lot of the main stem of the bay. And then when we put the two functions together, we can come up with sort of almost a bivariate type map here where Red would indicate both low growth and low shell building capacity. Kind of a green color is high of both. So you can imagine the green area is where a lot of the oyster livelihoods would be taking place. But there are some sort of mixed areas as well, particularly as you get kind of in the middle sections of the rivers that flow into Chesapeake Bay. You might have some nuance where you have maybe high growth but low shell building, or high shell building, low growth, like you see in the open ocean out here. And then with the spatialized interview methods, this is not the actual map that I was using. The screenshot on the right is the map that I was using for my interviews. But I do like that this picture shows that one of the folks I was interviewing was actually using a web-based map application to figure out some information about where to go to harvest on a given day. But essentially for these spatialized interviews, I had the map that I produced, the geovisualization tool, pulled up in front of people or over Zoom. And I was asking them to place points on the map, that's what all these markers are here, where they've observed a change that has affected oysters at some point in time. And then when they put a point on the map, a little questionnaire pops up where they can answer some questions, including please tell us a story about what happened. Can you characterize the change in different ways, like did it affect the survival or yield of the oysters? Did it affect the quality of the oysters, like shell strength or the speed of growth? How did people respond to the change? We asked a question about that. So did people increase their activities? Did they stop or decrease their activities? Or did they do something else? They could answer open-endedly on how they responded to whatever stories of change they're describing. And importantly, at the very end, we asked in their opinion what factors they think had an impact on shellfish. And this allows their local knowledge to really shine through in terms of what they see on the ground, being able to validate their best guesses for what the stressors were that affected the oysters, because we have this great biogeochemical model that we think we have these hypotheses about where the stressful areas are that people might see changes. But this activity allows us to kind of pinpoint and validate where, when, and what kind of changes are happening. So putting it all together, we're able to make a heat map of all the points that were collected from all across Chesapeake Bay, including parts of Maryland, eastern shore of Virginia, and the northern neck, Middle Peninsula, and Virginia Peninsula sides of Virginia. And we're going to be using this later in the project to integrate with our final public-facing web tool after we finish de-identifying and aggregating all the data. For now, I will just run through a couple of the preliminary results from the regression analysis that we did. And so we were testing the effect of the biogeochemistry conditions on aquaculture production. That's what this is showing you here. So the dark areas are where the effect of biogeochemistry is highest on production. The light areas are the places where that effect is lowest. And then this bar graph down in the lower left describes the relative importance of each of those individual stressors that fed into the prediction that produces this correlation coefficient map. So when we're looking at the whole system of Chesapeake Bay, we found that it was actually omega calcite saturation state that has the highest effect on aquaculture production. And if you notice in the abstract that I wrote for this talk, originally we were saying that salinity was the most important, followed by temperature and oxygen. But I actually just recently received the Maryland aquaculture production data, and that's what you're able to see on this map here. And by including that data, apparently omega calcite saturation state actually does show up as the #1 predictor for aquaculture production. So we did this type of test again using just the 2 species response functions we had. to predict aquaculture production. And in this case, again, the darkest areas are where species responses explain the most variance in production. And then between the two stressors, or the two predictors, I should say, scope of growth and net calcification, it's the scope of growth function that predicts aquaculture production the best. And perhaps that's intuitive. As the oysters are growing, people are harvesting, people are able to produce more pounds of oysters with larger growth. So perhaps that's what we might expect. But finally, I also tested the effect of exposure on production. And exposure in this context is related to the functions, but instead of using an instantaneous value at any one step in the time series, we were able to use an integral approach where we compared the duration of stressful conditions between two times, and did that for all of the times in the time series to compute an exposure metric that shows how far above or how far below the threshold for positive or negative tissue growth and shell building did those oysters experience. And when we looked at the effect of exposure on production, it was the net calcification exposure that affected production the most. And finally, to kind of tie it back into the interviews, the geovisualization tool that I'm showing here, it's the public facing view is still under development. So this is just screenshots from a private facing view that I have. But this is just zoomed in on a couple examples of some water areas that showed strong effects of the predictors like biogeochemistry on production. And then by zooming in, we were able to see the sort of time series metrics adjust to the local context that we're zoomed in on with the map. And so for this river system, which is a tributary of the Potomac River, it showed high effects of biogeochemical conditions on production. And when we looked at the importance of those, it was omega, calcite, and salinity. And to throw in a quote, someone was mentioning a few years ago, we had a strong freshet rainfall event that affected our production. And we can see a dip in growth and calcification, followed by a dip in production later. So this is one validation example that we have. Here's just another, I'll speed through it because I see Kirsten's camera come on. Another person I interviewed was describing how when freshwater discharge brings sediment into the bay, oysters could get covered up, and if no one is stewarding the reefs, then those oysters could cause a dead zone. And sure enough, we see drop in calcification and scope of growth, drop in production later, and the relative importance of the predictors for this example were salinity, the sediment or the total suspended solids, and the oxygen levels. So that matches the context that I found in the interviews. So just to speed through my conclusions here, we're describing freshwater discharge events as the type of event that OA operates in Chesapeake Bay, which affects production. This is maybe a little different from the West Coast, which is where my original research from my master's thesis was done, where we were finding that the upwelling effect has the largest impact on production on the West Coast. That's how OA operates in the Pacific Northwest. But in Chesapeake Bay, it seems like OA operates through these freshwater discharge events. So sure enough, production changes do follow those freshwater discharge events, particularly this one from 2018 to 2019 that interviews were reporting. The low salinity conditions are problematic for shell building, as we saw. This wasn't presented in my slides, but we do see that the largest farms appear to be the least affected, which we hypothesize may mean that the larger farms have more capacity for intensive adaptation to buffer those changes. We see the spatial variations in the effects, as we saw with those maps. Certain local impacts can have both a top-down and bottom-up explanation, as I showed in the previous slides. I mentioned this earlier, production might track with warmer temperatures, because that's maybe where an oysters are growing and when people are harvesting. But it's still uncertain right now how runoff and sediment affect production. But I'll leave it at that because I'm out of time. So here's a QR code to the current version of the geovisualization tool. The same link will have the updated version in just a few weeks. And yeah, here's my email. Thank you so much. I want to thank the NOAA Ocean Acidification Program and the MACAN Mid-Atlantic Coastal Acidification Network for inviting me to this webinar. Thank you all.
And thanks so much, Brian, for sharing your research today. We'll go ahead and make note of the QR codes and the products that are coming out of the research that both you and Sam have shared as well, and share that after the webinar with everybody too. So look for that coming, but thank you. Okay, we're going to go ahead and switch gears over to Catherine now, and she's going to talk some more about the biogeochemical model that underpins some of the work that Brian's doing and her own research with that model.
Cool, great. Thanks so much, Kirsten. Let me share my screen. Is everyone seeing that in full screen?
Yep.
Okay, perfect. Thank you. So yeah, I'm Catherine. I'm a second-year master's student at VIMS, and I work with Margie Friedrichs and Emily Rivest, as well as Mark Brush, Pierre St. Laurent, Sarah Blockman, Faye Daw, and Brian is also on the larger STAR project that he talked about. So I'm going to be presenting some of my work looking at climate change impacts on York River carbonate chemistry and oyster growth. And I want to acknowledge funding sources from the NOAA Ocean Acidification Program, as well as VIMS academic studies. So some background about the study species, the eastern oyster. The eastern oyster is an ecosystem engineer or keystone species in the Chesapeake Bay. So they provide a lot of really valuable ecosystem services. They are filter feeders, so they filter the water and help with water clarity. They also help with benthic pelagic coupling and denitrification. And through building 3D reef structures, they help with shoreline stabilization as well. They're also very valuable economically. As we just heard from Brian, Virginia aquaculture is really big industry. And also through those reef structures they provide habitat for other really valuable species like blue crabs and striped bass, which are huge commercial fisheries in the Chesapeake Bay. But coastal acidification is sort of threatening oysters right now in the bay. Sure, most people attending this know about ocean acidification, but I'll go over the processes on the right. Here we're looking at the equations of ocean acidification. As the ocean absorbs excess anthropogenic CO2, it'll react with seawater, form carbonic acid, which will dissociate into free hydrogen ions and bicarbonate. The free hydrogen ions will react with free carbonate ions to form more bicarbonate. And this becomes a problem for calcifying organisms, as it means that less carbonate is available in the water column for them to build their calcium carbonate shells. And then looking on the left, in coastal and estuarine systems, this whole process gets exacerbated because freshwater has a lowered pH and a lower buffering capacity or total alkalinity. So that means it can't resist changes in pH, and we see really short-term variations in pH in these systems. Freshwater also has a lower amount of dissolved inorganic carbon, so that means there'll be less carbonate. And freshwater has a much lower concentration of calcium ions than in saltwater. So all of those issues contribute to coastal acidification threatening calcifying organisms. And this is important for the eastern oyster since their shells are made up of calcium carbonate. Specifically, they use two forms of calcium carbonate. So they use aragonite to build their larval shells and then calcite to build their adult shells. I'll be focusing on calcite since my work is focused on adult oysters. And omega calcite is just that measure of the ambient amount of calcium and carbonate available in the water column. In some prior work by members of our project group, including Emily Rivest and Mark Brush, they found that the eastern oyster has a calcification threshold of 0.93. So that means when omega calcite is above 0.93, they'll experience net calcification, and it'll be energetically favorable for them to build their shells. And then when it's below that threshold, they'll experience net dissolution. And so we have a good hold on how acidification and carbonate chemistry influence oyster shell growth, but obviously climate change, there's many different changes occurring at the same time. And so just one co-stressor that I'm focused on in this work today is atmospheric warming. The ocean absorbs about 93% of anthropogenic heat, and the Chesapeake Bay is also located off the North Atlantic, or the Mid-Atlantic, and that area has historically warmed faster than the global ocean. So there's a lot of warming and acidification happening in this region. And the effects of warming on oysters are a little less direct and clear than the effects of acidification. Warming will lead to increased respiration rates, which will cause reductions in pH and omega calcite in areas where the surface waters and bottom waters will be decoupled. So this might affect carbonate chemistry and reduce calcification, but at the same time, warming also leads to faster growth rates. So it's a little less clear how that will affect oysters. And that brings me to my research question: how will future coastal acidification and warming, both separately and together, influence oyster shell growth in the bay? So to get into my methods, I'll be examining this question in the lower Virginia tributaries of the Chesapeake Bay. So the study region is pictured here on the right. And this is a really extensive region for aquaculture and restoration. If you see all the little black dots on this map here, those represent private oyster leases. So you can see they're everywhere, particularly along the coasts. And this doesn't include areas of restoration or public oyster lands. This is also a really high management interest area. Just last year, NOAA announced the Middle Peninsula, which includes the York River, the Mobjack Bay, and the Piankatank River region, as a habitat focus area. So they're putting a lot of resources towards restoration and research into the effects of oyster restoration and wetland restoration and other management issues. And then to answer this question, I'm using the model that Brian alluded to earlier. The version of the model of ROMS-ECB, this is a 3D estuarine-carbon-biogeochemistry model. And the version of the model I'm using is our highest resolution. It's 120 meter horizontal resolution. So that means that we can look at really shallow coastal areas with much higher resolution and accuracy than in our larger whole bay models. And the carbonate chemistry variables in this model are dissolved inorganic carbon and total alkalinity. And then we can calculate omega calcite from those. The other model that we have is eco-oyster. So this is an oyster bioenergetics model, and it's been embedded within ROMS. So together, the product is ROMS-ECBO, and this will simulate daily growth of a diploid and triploid oyster, while it's also simulating all those environmental conditions in the bay. And the schematic on the right is demonstrating very simply how eco-oyster works. We have many different growth functions that are based off of environmental variables, and they simulate the growth of somatic tissue, reproductive tissue, and shell. And in this work, I did 4 different model runs. So first, I just have my reference run, which was representing 2016. And so it's represents realistic conditions. It's been compared with observations throughout the bay. And then I did 3 different experimental runs looking at the future, so mid-century of approximately 2066. So it's 50 years ahead of our reference run. My first run is a future atmospheric CO2 run, which includes forcings of atmospheric CO2 from the IPCC report, the RCP 8.5 scenario, which is the worst case scenario or business as usual scenario. And so ambient atmospheric CO2 concentration is about 646 ppm in that run. The next run I did is a future atmospheric temperature run. And so to calculate the change in temperature, we used downscaled atmospheric temperature data, and I calculated a 50-year difference over 2 time periods centered on 2050 and 2000. And so since I used downscaled data, the change in temperature is different throughout the year and throughout the model domain. But just to get an idea, I took an average across the whole year. And the average is about an increase in 2.37 degrees Celsius. And my final run is a combined run. So it has the forcings of future atmospheric CO2 and temperature to see how they interact with one another to affect carbonate chemistry and oyster growth. Now getting into my results. All the plots on this slide are going to represent calcite saturation state, average from May to September. So that's the main oyster growing season. And the plot on the left, the plot we see here is showing 2016 average values over that time. And you can see that the omega calcite saturation trends follows what we saw with salinity and what Brian was showing, where we see higher omega calcite in the higher salinity areas, so further down the tributaries, and also in those shallower areas of the tributaries. And the next three plots I'm going to show are showing a difference in that average value between the future run and the 2016 run. So the color bar now, all the blue will mean that there's a decrease in omega calcite, and if it's red, that means there'll be an increase in omega calcite. And as expected, we're seeing a decrease occurring across the board with future atmospheric CO2, particularly in some of these really shallow tidal tributaries. We're seeing a lot of change, less change in the more saline and higher alkalinity waters of the main stem. With temperature, excuse me, we see the opposite effect. This is largely due to an increase in photosynthesis rates that are occurring with higher temperatures. Since in those really shallow areas, we're going to see increased primary production, but there's enough mixing that the effects of respiration aren't overtaking the shallow areas. And then in those deeper areas along the main stem, that's where we might see the effects of increased respiration affecting omega calcite more than photosynthesis. But when we have a combined run, you can see that the effects of temperature sort of are completely overtaken by the effects of omega calcite. We do see a reduction in those really dark regions along the edges of the Rappahannock and the York. So temperature is helping to offset the effects on carbonate chemistry, but not enough to outweigh them. On this slide, what we're looking at is the same scenarios, but now instead of a May to September average, we're going to have shell weight after 1.5 years. So I ran the model for 1 year and the oysters start off at 6 months. And this is just showing what the weight is at the end of the year across the model domain. And we see that in 2016, shell weight really follows that calcite trend, where we're seeing really high shell growth in the lower parts of the tributaries along the shoals and along the main stem, where it's really high saline water and high omega calcite. And next, getting into CO2 and temperature, we see pretty much the same trends that we saw with omega calcite, where under future CO2, we're seeing a reduction in shell growth due to that reduced omega calcite. And then under future temperature, both the increase in omega calcite and the increase in growth rates from higher, warmer temperatures cause an increase in shell growth. But when we get to the combined run, it's a little different than what we saw with omega calcite. We're seeing how the effects of climate change are really spatially variable in the bay. So we still have dark blue slowing growth in the upper ends of the Rappahannock and the York. The reason that it's white all the way at the top is just because there weren't oysters there to begin with. So it's just a net change of 0. But we're seeing that as you move down the tributaries and it's low salinity, those are the regions that we're seeing that decrease. And then along the main stem and even further down the Rappahannock and York, we're actually seeing an increase in shell weight under future conditions. So that's really interesting to us to just see that there definitely are regions where we're going to see much more vulnerability to climate change, and then other regions where there's a lot of resilience where they might even benefit from climate change. And so really quickly, my final slide I want to show is just this figure is demonstrating this calcification threshold that I mentioned earlier. So I calculated across the model domain how many days in a year each grid cell spends below that calcification threshold. And then I took a difference between the future combined run and the reference run. And what we're seeing here is that result. So basically where it's going to be crossing that threshold the soonest and the most is probably the regions that are going to be most vulnerable. And so similar to what we saw before, we're seeing that change occurring sort of midway up the Rappahannock and the York River, and then also further down the river, just along those shallow shoals. And if you recall from the salinity plot that I showed earlier that had all the private oyster leases, If I overlay those on top of this figure, we can see that a lot of these regions that have a lot of aquaculture, these shallow regions and the areas along the edges of the tributaries are also coinciding with some of the regions that are going to be changing faster. And if you look at the color bar here, some of this change is really dramatic, like up where it's red, up and further up the tributaries, that's up to 100 days more that these oysters will essentially not be building shell because omega calcite will be too low. So this means that aquaculture practice could really be at risk in these regions. So just some conclusions and implications from this. As we saw, the effects of climate change in the bay are going to be very spatially variable. As well, acidification will decrease shell growth, but warming will have a stronger effect in some areas and offset it throughout the bay. And as a result, aquaculture and restoration in this region may be threatened. I also just want to note that my future work will also include the effects of sea level rise and the effects of land use change. So looking at how combined all of these different effects will influence the region. So that's all I have. Thank you so much. My email's here and I'll answer questions after this.
Thanks so much, Catherine. We're going to switch over to Annie now to talk about, we've been looking at a lot of modeling approaches and now we're going to look at a genetics approach. So we'll totally shift gears and learn some more about that as well. If you do have questions, please remember to put them in the question and answer box because we're going to do all the questions after Annie's done presenting today. Thank you.
All right. Hi, everyone. As Kirsten said, my name is Annie Schatz. I'm a PhD candidate at VIMS. And we will be moving away from modeling and into some experimental physiology work. So slides look okay?
Yep.
Awesome. Wonderful. So my research today that I'll be sharing is looking into these two different types of eastern oysters and trying to understand how their larvae may respond physiologically to climate change. So Catherine did a great job of telling you why we care about oysters and the importance that they have in our local environments as well as to our economy. But there are two different types of oysters that play these roles. So the first is the one that you're probably more familiar with, your wild reef-forming oyster. And then the second is your selectively bred oyster. So these oysters have been chosen and bred over several generations for specific traits like fast growth, disease resistance that are beneficial to the aquaculture industry. And so it's this selection for these specific traits as to why I think that they actually may be more equipped for handling stress. And that's because with the selection for these traits over time, they may have a different way of allocating their energy resource compared to our wild oysters. And therefore, they may actually have a bit more capability or more energy to allocate towards that external stress. But both of these types of oysters depend on the success of their already vulnerable larval life stage. So the larval stage, if you're not familiar with the oyster life cycle, is actually about 2 weeks and they are mobile. They live in the water column. And during this time, they're undergoing all these developmental processes, and those processes take up the majority of their energy, leaving very little left over to handle external stress. So we're interested in understanding how the stress may be combated at the larval stage when they already have such little energy. And so I used this bioenergetic framework that was created by Sokolova et al. In 2012, where as an oyster or an organism begins to experience more and more stress, they begin to prioritize most of their energy towards those processes that are necessary for survival. And this in turn decreases the energy left over, or what I've referred to and labeled here as the aerobic scope, which is the energy that supports all of these other important aspects of an organism's biology, like growth and long-term energy storage. So using this framework within the context of the little energy that larvae already have, I wanted to understand, under the context of environmental stressors within the Chesapeake Bay, how these oysters may respond. So Catherine did a great job as well, explaining acidification within the Chesapeake Bay. We already experience conditions that are well below the IPCC's 2100 prediction for the open ocean. And then I'm also, similar to Catherine's model, putting this into context of multiple climate change stressors, since we know that that is the reality of climate change for these organisms. So altogether, the objective for this study was to understand how these two types of larval eastern oysters, so wild and those from selectively bred aquaculture broodstock, may respond physiologically to the combination of ocean acidification and ocean warming. So just briefly, how we did this, it was conducted over 2 years, and we strip spawned adult oysters, and immediately after fertilization, the embryos were exposed to a combination of 2 different temperature and pH conditions that represent our current average ambient conditions and our current extreme conditions that these oysters already experience within the tributaries of the Chesapeake Bay, which we anticipate becoming more frequently experienced in the future. This gave me a total of 4 treatments that allowed me to assess both the individual effects of these stressors as well as the combined effect. So bringing it back to that bioenergetic framework, I took a bunch of physiological samples, but these are the ones that I'll be sharing with you today. The one that I want to point out really quickly is for the cellular maintenance category. I looked at lipid peroxidation, which is a metric for oxidative stress damage, and oxidative stress. That way we can kind of tie it to how much oxidative stress or cellular stress they may be experiencing, and therefore how much energy they may need to allocate to that cellular maintenance category. So before diving in, I'm just going to orient you to this graph very quickly. On the left-hand side for the rest of the talk will always be the response from our wild oyster larvae. And on the right-hand side will always be the response from our selectively bred larvae. Here we have along the x-axis our 4 different treatment conditions, which are color-coded and will be the same throughout the talk. So control in the dark purple, temperature, the warming only treatment in the light purple, the acidification only treatment in the light green, and the combination of warming and acidification in the multis treatment in the darker green on the far right-hand side. Along the y-axis, we have our physiological response. So here I'm showing you cumulative survival. So throughout the entire experiment, which was ended about halfway through the larval stage, so around 8 days post-fertilization, what we found for survival that relates specifically to the difference in the type of oyster was this response to conditions of acidification. So we found that our wild larvae are more lethally impacted by acidification compared to our selectively bred, where you can see there's very little difference amongst all of the environmental conditions that they experienced in terms of their survival, which is what I had hypothesized. So now moving on to our proxy for tissue growth, which we use total protein for. Here along the x-axis, I now have the age in days post-fertilization or our sampling time points. So the one thing that I want to draw your attention to first is when comparing our acidification treatment to control conditions, we found that our selectively bred oysters, as we anticipated, are not really affected by acidification alone, while our wild oysters do experience this decline in their tissue growth for the first few days of their larval life. When we look at shell length for comparison, we see a pretty similar pattern in these 2 types as well. So you see that in our wild oysters, we do have this difference between the control being impacted by acidification. And the difference here with our selectively bred is that initially there actually is a negative impact of acidification on their shell growth, which you would anticipate, but they quickly make up for that. And then another thing I want to draw your attention to is that while there was this initial negative impact on wild larval shell and tissue growth, they do also seem to bounce back from that towards the middle portion of their larval life stage. So when we add in the effect of the co-stressor of warming, we see that it's very beneficial actually to our wild oysters. Their total protein is not very different from this really faded control line, and it is significantly more, it's significantly greater in our co-stressor treatment compared to the acidification only. That positive benefit is actually not as drastic in our selectively bred oysters, which I thought was interesting. And eventually too, towards that middle portion of the larval life stage, we actually start to see it become almost a negative effect in their tissue growth. When we look at their shell length for comparison, we see again that added co-stressor of warming seems to ameliorate any of those negative effects of acidification for our wild oysters, but compared to the selectively bred, again, we don't really see that great of a benefit of that co-stressor of warming on shell growth. So now moving into lipid damage, which again, we use as a proxy for oxidative stress or cellular stress experience. There are a couple of things that we found with some of these results, but the one that I want to draw your attention to is that over time, for those first few days of the larval life stage, we actually see that our wild oysters have much more lipid damage and therefore more oxidative stress experience compared to our selectively bred oysters, which seem to have less lipid damage accumulation and seem to be less stressed overall. But what's interesting is that this does switch towards that middle portion of the larval life stage. We start to get this tapering off in our wild larvae of that lipid damage, while our selectively bred oysters continue to accumulate that lipid damage and are seemingly more stressed now compared to our wild oysters. So putting this all together, what we're seeing as a quick review is that our wild larvae seem to be lethally affected in terms of their survival with acidification compared to our selectively bred oysters, who don't seem to be as affected or impacted with their survival through time. And when we look at the aerobic scope, or just generally how their physiology is doing within the context of this bioenergetic framework, we see that in those first few days of the larval life stage, it's kind of what you would anticipate, that our selectively bred oysters seem to be better off and are more able and capable of handling those environmental stress conditions. We found that their tissue growth and their shell growth was relatively unaffected by acidification and was not really affected negatively by the co-stressor of warming. Similarly, they had less lipid damage from oxidative stress experience compared to our wild oysters, where we saw that negative effect on their tissue and shell growth from acidification, and that higher lipid damage comparatively. But towards that middle portion of the larval stage, we start to see this pattern flip. So we started to see that the growth of our selectively bred oysters with respect to their tissue started to decline when they experienced the two stressors together. Their lipid damage and experience of oxidative stress seems to increase compared to our wild oysters, who generally tended to taper off in that lipid damage. So they seem to be responding or maybe acclimatizing to that stress, and their growth seemed to recover from that negative impact of acidification. So overall, it seems like from these results, we're starting to see evidence that our wild oyster larvae in the long run may actually be better able to withstand environmental stress or multiple environmental stressors compared to our selectively bred larvae, which may be potentially negatively impacted, especially as they get close to that metamorphic time period, which is very stressful for them. So I have lots of people to thank for the help with this project, as well as my funding sources. And there is my contact information if you have any questions after this. So with that, I'll say thank you.
All right, thank you very much to all our speakers. Just give a final reminder, if you have any questions, to please type them in the Q&A box now, and I can ask them to our speakers. But to start us off, first question here is for you, Brian. Could you tell us more about the public-facing web tool that you mentioned you're planning to develop?
Yeah, so it's a web map that you can explore a lot of those data layers that it's presenting. I'm going to be posting the regression analyses on that public-facing map in time. There's just a couple of little tweaks I'm making to the design, but you should be able to actually go in and play around with the drop-down menus I have so that you can look at all of the different combinations of regression tests and predictors and predictands that I was using. But yeah, it'll just be there on the web. It's available for everyone to see.
Great, thanks. For our next question, for Catherine, does your model also take into account potential fluctuation from storm events and how that may also reduce days oysters are able to grow?
So there's variations in precipitation throughout the year. So like, for example, 2018 was a really wet year. So if we look at results from then, you can see the effects of the really high amount of precipitation and the changes in salinity. I don't think it has like every short-term major storm, but it does have that overall climatology. And we can also, members of my lab, Faye has done experiments where he has sort of forced a high precipitation event into the model and looked at the effects on carbonate chemistry.
Great, thanks. So we'll go our next one to Annie. Do you have an understanding of why kind of around that halfway point you started to see that switch in cellular stress between wild and selectively bred oysters and where that may be coming from?
Yeah, that's a really good question. I have been pondering that myself as I am writing this up for my dissertation defense. Some thoughts that I've had are something with their lipid stores. The lipid data is, I do have some lipid data. It's a little bit confusing and muddled. What I am seeing from the lipid data is different patterns of utilization. So overall, the selectively bred oysters have less lipid stores than our wild counterparts. So I don't know if that means that they've been sublethally stressed that entire time and that it's just manifesting later on in the larval stage, and that they've been utilizing those lipid stores instead of being able to store them throughout time. And so maybe that is leading to that decline, but it's definitely something I'm working on interpreting, and hopefully we'll have a better answer soon.
Great. So our next question here is addressed to both Brian and Catherine. Do you know if your methods could also be applied to some of the hard clam aquaculture that happens along the bay side of the eastern shore and in the southern parts of Chesapeake Bay, or if your results may be representative of kind of what you could also expect in that species as well, if you know?
Catherine, you want to go first?
Yeah, I think for the eco oyster model, those formulations are based off of an experiment done with an eastern oyster. So like for the actually simulating oyster growth or clam growth, I think you would need to do an experiment that would be able to like build that model. But if you knew approximately what the thresholds were for clam aquaculture, then like all the environmental variables would be the same.
And to piggyback off of that, the production data that I was working with from the Virginia Marine Resource Commission, it does also include clam aquaculture. I just didn't use it in the analysis I was presenting because of what Catherine mentioned, that we're working with oyster response functions, not clam.
Great, thank you. Another question here for Brian. What have farmers/watermen cited as the biggest change that they have noticed over time in oyster production? Has it been shell length or total amount of meat or overall size or something else?
Yeah, that's a great question. As far as like the impact of the oysters, they are reporting issues with the feeding rates. So in some cases, there's stalked ciliates or a type of protist that can attach to the oyster shells in their really early juvenile stages and can kind of scoop out some of the food that the oyster would otherwise be eating for itself. So that can be an issue that was reported kind of all over the bay. But freshwater events, like I was mentioning, came up a lot, as well as some other sort of social stressors unrelated to the biogeochemistry, but a lot of sort of livelihood diversification factors like somebody, I'll just say laundering money came up a little bit. So, and engagement in illicit activities to the point where it was actually harming other oyster farmers in the nearby area. So I'll leave it at that, but it's just to say there are a whole combination of stressors that can impact oyster production, both environmentally and socially.
Great, yeah, thanks for that insight. Our next question here is for Annie. Did you grow your oysters past that halfway point that you showed, and do you still see persistent effects beyond that point?
Good question. So the original intent was to continue both of these populations into the first month of their juvenile stage. But what ended up happening was right around the time that they start to become pre-metamorphic, they started to, and it's an interesting result in and of itself, both types started to crash in the multi-treatment, but for some reason our wild oysters crashed even more so in some of the other stress treatments. So to a point that I wasn't able to continue on with the experiment beyond metamorphosis. But I did with the with one of the populations, I was able to continue it further into that month after their juvenile, after metamorphosis, so the first month of their juvenile stage. And I am looking into those data probably starting next week a little bit more. So I look forward to that and get back to you.
Great. Okay, so the last question I see here, I think Brian and Catherine, you may both be able to speak to this. It's in reference to kind of the sites of oyster production you showed. Do you have a good idea of like what percentage of oyster growers are producing oysters in the regions of the bay that you both identified as kind of the most threatened, or where they would see the greatest decrease in oyster growth in the future? Or is that something you haven't quantified?
Most of those areas are growing oysters. I don't know if there's like an exact percentage, but the majority of the leases that Catherine was showing on her slides are all oyster producing leases. I will say it's required by the state to show that harvest is being done, which is what keeps people maintaining their leases with the states. So although the production data is showing that production is happening in all of those places, as I mentioned in the last answer, if production data is being faked or other issues are happening, then perhaps there's a smaller percentage than what we think that are actually growing oysters in every lease, but more evidence would be needed for that.
Okay, great. That's all the questions that I have here. So I'll turn it back over to you, Kirsten, to wrap us up.
Thanks so much, Anthony, for moderating this session today. And I also want to give a big shout out to Anthony. He's been a MACAN OA fellow for the last couple of years, and he's been super helpful with our webinar series. And this is his final webinar with us, so we're really going to miss him, but we appreciate all that you've done over the past couple of years and wish you luck as you head on into finishing your dissertation and moving forward. So thank you. And
yeah, thank you. I'll need it.
Yeah, and thank you so much, Annie, Catherine, Brian, and Sam, who had to leave a little early today for joining us and sharing all the great work you guys are doing. It's really interesting to see. It's interesting to see the breadth of work that's going on, especially in the Chesapeake Bay area too. So thank you for sharing that with us. We're looking forward to seeing those tools and products as they're developed as well. And just one more shameless plug, you know, if you missed our webinars, the recordings are available on our website on macan.org. And hopefully we'll see many of you at our State of the Science workshop in September. So thanks so much.
Thank you. Bye.
Thanks.